Hello, I'm Kasane YASUOKA, and today I would like to talk about expression and localization of ACE2 protein in the primary culture of human airway epithelial cells. So my theme is related to SARS-CoV-2, so I will explain briefly about SARS-CoV-2 and its entry process. Severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, is a causative agent of ongoing pandemic disease COVID-19. The virus is an enveloped and single-stranded RNA virus, and it contains spike protein from the surface which is responsible for invasion. For the entry process, first spike protein recognizes membrane-bound protein from the surface of the cells known as angiotensin converting enzyme 2, ACE2. After spike protein binds to ACE2, spike protein gets cleaved by some protease. First, between S1 and S2 domain of spike protein was cleaved by furin protease. And next, the S2 domain was cleaved by TMPRSS2. By these steps, the membrane fusion domain is exposed and activated so that membrane fusion occurred and virus entry to our body. Next, I would like to talk about ACE2 briefly. ACE2 consists of 805 amino acid, and it is type 1 integral membrane protein. The N-terminal segments exist in extracellular part, and C-terminal segments exist in intracellular part. And it contains 7 N-linked glycosylation sites in their structure. ACE2 is a major component of Renin Angiotensin Aldosterone system, RAAS. In this system, angiotensin converting enzyme, ACE, converts angiotensin 1 into angiotensin 2. And angiotensin 2 binds to angiotensin receptor that elicits vasoconstriction or proliferation or hypertrophic effect. On the other hand, ACE2 converts angiotensin 1 and angiotensin 2 into angiotensin 1 to 9 and 1 to 7 respectively. Angiotensin 1 to 7 binds to the MAS1 receptor that elicits vasodilation or anti-hypertrophic effect or anti-proliferation. So it is intriguing that ACE2 have a multiple function. These days, ACE2 isoform of splicing variant, which lacks the N-terminus, this is the image, is reported in airway epithelium. So next, I would like to talk about airway epithelium. Airway epithelia consists of ciliated cells and secretory cells. Ciliated cells are hair-like organelles which is responsible for mucociliary clearance, and it is the primary defense mechanism in the airway. Airway epithelium is covered with thin layer of mucus that traps and removes pathogens, including SARS-CoV-2 or particles from inhaled air by ciliary movement. These days, immunofluorescence staining studies have shown expression of ACE2 in ciliated cells of airway epithelium, but the reported subcellular localization of ACE2 varies depending on the antibodies. In addition, by Western blotting and immunofluorescence, the splicing variant of ACE2, which lacks N-terminus, as I said in introduction, was reported, but however, the subcellular localization of ACE2, including its variants in ciliated cells, is still unclear. In this study, we investigated ACE2 protein expression and localization using multiple anti-ACE2 antibodies by Western blotting and immunofluorescence. In this study, I used two kinds of airway epithelial cells. First, normal human bronchial epithelial cells, NHBE. NHBE was purchased from Lonza company. And next, I used ciliated human nasal epithelial cells, CHNAC. And these cells were provided from Kyoto Prefectural University of Medicine. I would like to take this opportunity to say thanks to Dr. Yasuda and Dr. Okamoto for providing us the cells. And these cells were cultured by the air-liquid interface LLI method. LLI is a method to differentiate cells into ciliated cells. For the process, first the cells were cultured in the permeable support filter transwell, and after cell reached confluence, the medium in the top chamber was removed and the apical side of cells were exposed to the air. This state is called air-liquid interface, and it resembled the environment of the airway, so the cells differentiated to ciliated cells. So I would like to move on to the result. This is the result of immunohistochemistry of NHBE and CHNECs after the treatment of LLI. The day 0 shows the starting day of LLI, and I made a sample chronologically. I stained with acetylated tubulin in red, which is a marker of cilia. As a result, the cell detected cilia clearly from about 7 days, and it's developed, and in 21 days, the cell surface were full covered by cilia. So next, I made the lysate samples of each days, and I did a Western blotting to detect the ACE2 protein expression. In this study, I used 3 kinds of ACE2 antibodies. First, HPA00288, it recognizes the N-terminus of the ACE2. And next, I use AF933, it recognizes the middle part of ACE2, including ectodomain. And last, I use AB15348, which recognizes the C-terminus of the ACE2. So this is a result of ACE2 expression using each 3 antibodies. Number 0 shows the starting day of LLI, and I made a sample chronologically. Day 0, the ACE2 expression was scarcely observed, but it's markedly increased by 7 days, and it's remained constant in high level. And the band patterns were different among antibodies. Antibodies recognized by AF933 and HPA00288, the band detected at 135 and 100 kilodalton. On the other hand, AB15348, which recognizes C-terminus of the ACE2, detected the bands at 135 kilodalton in single bands, and it also detected the bands at 50 kilodalton too. This 50 kilodalton of ACE2 are corresponded to the splicing variants of ACE2, so it seems to represent the ACE2 processed of splicing variant. And other bands are all detected higher molecular weight than expected, 97 kilodalton. And as I said before, ACE2 contain glycosylation site in their structure, so next I did the N-glycosidase F treatment. In the left side, it's the before the treatment, and in the right side, it's after the treatment. The bands detected at 135 and 100 kilodalton before the treatment of N-glycosidase F was shifted to 90 kilodalton and 70 kilodalton, respectively. So from these results, it was suggested that ACE2 protein represents 3 forms in the lysate samples. First is the full-length ACE2 of molecular weight of 135, which both contains N-terminus and C-terminus. And next is the short ACE2 of 100 kilodalton, which contains N-terminus, but it lacks C-terminus because it was not detected by AB15348 antibodies. And at last, the ACE2 of splicing variant of 50 kilodalton, which contains C-terminus, but it lacks N-terminus. So next, I focused on the short ACE2 of 100 kilodalton, and I hypothesized that it is one affected by protease TMPRSS2. So next, I cultured the NHB cells with chemostat, which is a TMPRSS2 inhibitor. And the lysate was blotted by ACE2 antibodies. 100 kilodalton observed in control lysate was disappeared in the lysate of cultured with chemostat. So it was suggested that 100 kilodalton of ACE2 represents ACE2 processed by TMPRSS2. So next, I detected the furin and TMPRSS2 protein expression too. Both proteins were detected in NHBE and CHNEC, and unlike ACE2, the expression was observed even before 0 days, and the expression level were unchanged before and after LLI. So at last, I did immunofluorescence to know the localization of ACE2 expression. The ACE2 signals observed in AF933 and HBA00028 in green signals were co-localized with deciliated cells in red almost completely. On the other hand, AB15348, which recognizes C-terminus, the ACE2 signals in green were located as apical membrane as well as intercellular part. These signals were disappeared in the presence of antigen peptide. So from this result, it was suggested that full-length ACE2 and C-terminal deficient short ACE2 were mainly localized at cilia, whereas the short ACE2 of splicing variants were mainly localized at apical membrane of the cells as well as intercellular compartment. And it's all for my results, and in conclusions, in the primary culture of human bronchial and nasal epithelial cells, expression of the ACE2 protein was increased by 7 days in LLI culture. And ACE2 has at least 3 forms: full-length ACE2, N-terminal deficient short ACE2, and C-terminal deficient short ACE2. The C-terminal deficient short ACE2 seems to represent ACE2 processed by TMPRSS2. Full-length and C-terminal deficient short ACE2 are localized in the cilia, whereas N-terminal deficient short ACE2 localizes in apical membrane. And at last, ACE2, TMPRSS2, furin, which are important for SARS-CoV-2 infections, were expressed. Therefore, bronchial and nasal epithelial cells are suitable for SARS-CoV-2 infection, and it is important for the important entry routes for SARS-CoV-2 infection. So this is all for my presentation. This is acknowledgement. Thank you very much.